like to get today's session started, and we have a very well-known affiliate of FABA with us, Dr. Anu Acharya. I would like to quickly introduce Dr. Anu to all of you before I hand it over to her. Dr. Anu Acharya is in fact a serial entrepreneur. She's a columnist, she's a poet, and she's a charismatic speaker. She's also the founder and the CEO of Mapmygenome, which has done phenomenal work in the field of personal genomics in India. And she also launched something very interesting called the Genomepatri in 2013. She co-founded and pioneered her first venture called Ocimum Biosolutions. This was between 2000 and 2013. Through very innovative bioinformatics and LIMS solution, global acquisitions, awards, and fundraisers, which is something that India really sees a dearth of. She has a number of awards and honors to her name. She has been recently awarded with the She Is 75 Women in STEM award. She has been awarded with the WTI award by the Niti Aayog Kendra, Women Entrepreneur of the Year award by Indian Express. This was this year itself. The Woman Entrepreneur of the Year by Z Business as well. She has a special recognition amongst the leaders by the Indian Express, recognized by the Indian Express. She also received the Pride of Telangana Award, and we're very proud of you as well, Dr. Acharya, in 2018. Triple IT Kharagpur also distinguished her as an awarded her as a distinguished alumnus. She went on to receive the Young Leaders Forum of British High Council Award, Young Global Leader of World Economic Forum. This is from the class of 2011. She presently serves on a number of governing bodies of the NIBMG, Triple IT Hyderabad, and the We Hub. She is also on the advisory board at Action for India and KIIT. She is on the board of mentors for the Ivy Cap Ventures, a member of the CII National Committee on Biotechnology, the committee member of HIMSS Asia Pacific India Chapter, NASCOM Executive Council member. And with that very wonderful set of introductions and very nice set of accolades to have in your name, I'm very happy to introduce Dr. Anu Acharya to our audience. Thank you so much for being here today.
Thank you so much for the kind introduction, Dr. Meenakshi and Dr. Ramesh. And also always a pleasure to see Professor Redana over here as well. So I thought that it might be, FABA, I remember from the time that it was first conceived, and it has always been a pleasure to see how much it has grown. And I think what we're doing now in the field of training and bridging that gap between industry and students, I think that is an area that is hugely appreciated. I think we badly need that. I think there are very few programs that are actually aiming to do that. I think one of them is BIRAC, the one that the DBT has done. But I think there are many more that we need to do to be able to bridge that, because clearly I think there are many students who are aspiring to be part of the industry and part of the growth, and we are not able to necessarily find the right talent. And I think it's mostly about connecting the dots, as we would say. So today I thought I will take you across a little bit of my journey, and I think the idea really is to help people understand that, and maybe if I can get access to. So I thought I'll maybe take everyone through a little bit of my journey to show that, sometimes I think, especially when you are younger, I think we all think that our life is completely going to be panned out the way we hope it will. Now, one thing I want to show is that sometimes what you study and sometimes what you end up doing might be two totally different things. But I think what we first look at, what we first attempt to get to, I think that sort of determines where our journey will eventually go to. So I always mention that I was born in Bikaner. My genes are all from Rajasthan. But I grew up in Bengal, so I think half of the epigenetics all came from Bengal. But I also went to IIT Kharagpur, University of Illinois, and Harvard Kennedy as part of my education in different areas. Some of it is in public policy, some of it was in information systems and in physics and so on. So I think I did all of that. And my first journey in genomics started in the year 2000. And at that point of time, I think the Human Genome Project had just about been released. in 2000, 2001. And it was still something that was very, very new. The whole genomics, bioinformatics field was quite new at that point of time. But I think it sounded like an interesting problem or an interesting challenge for us to solve. And that's why we got on to being able to create something like Ocimum Biosolutions. But at that point, it was more about leaving the US, coming back to India and to be able to start that. What was interesting at that point was that it is very, very hard to find anybody who had even the basic knowledge from a bioinformatics point of view. And so it was hard for us to get talent to be able to at least even get the process started. So we had to look at a lot of the global talent that was available. We also had several other challenges in terms of training. And so we working with other universities in the US to be able to bridge those gaps that we had at that point of time. But as we went along, I think over the years, we did 3 acquisitions, one in Germany, one in Netherlands, and one in the US. And all of these put together, I think we became one of the largest genomic services players worldwide. But at the same time, I think was an interesting point of time in terms of what really happened. And these are things that are not in your control. And at that point of time, we had the financial crisis of 2008. And I think that sort of changed a little bit of how things were going to be in the pharmaceutical world, changing the way how it was in the biotech world, and also just in terms of just the amount of funding that you could potentially get to be able to grow, especially like what we were thinking about was through acquisitions. But there was also many other changes that were happening across the globe. And one of them was that the world was moving towards moving genomics towards the clinic. And the other thing was, of course, that a lot more people realized that the power or the value that you can get from genomics being given to that of a consumer. And I think we started looking at some of those, and we started looking at how do we bring this to the Indian market. So around 2013, we launched our first product in Mapmygenome, and we aptly called it Genomepatri. And as some of the professors recently told me in another conference that I was speaking at, that Genome and Genome actually have the same word origin. And so it was interesting that the name that we gave ultimately was a perfectly fine name. But over the years, I think we had built this platform for a consumer where people can order online and get all of that. We also added to that also clinical sequencing, NGS, and exome sequencing to that mix. And over the years, I think we came across another interesting global crisis. And this time, the global crisis, we were able to help out a lot more because we already had been doing PCR and other such molecular biology for the last 22 years. And so it was 20 years. And so it was easy for us to be able to become, one of bigger contributors in the space of helping in the COVID space, whether it was in COVID testing, whether it was in looking at a lot of the seroprevalence studies. And as a result of all of that, because we had people coming into our labs quite regularly, I think we also realized there was some value that we could do to be able to connect some of the other dots that were there. That was, for instance, the biochemistry. So we launched some biochemistry tests as well very early, not very long ago, about 6 to 8 months ago in Hyderabad. And we've also launched products that are there in Bangalore and also in Delhi as well. So I think that's what we're seeing. And one of the areas that we want to get to is to be able to ultimately be able to find a way to sort of combine the data types to sort of help people understand and optimize their lives better. So I wanted to bring back, and then especially I think this is valuable if there are students in the audience, that one of the things that we can see is that sometimes we find that science fiction or movies actually can think about things even better or faster or earlier than maybe even scientists can get there. And an example of that was this movie called Gattaca. And as most of you who are in the FABA world will probably realize, these are all ACTGs. And this was done in 1997. And it starts off, the trailer starts off with in the not too distant future, a single drop of blood or a single drop of saliva can tell you who you are, who you can marry, and so on. Now, this was obviously not the most, positive movies in some sense, and it was in some sense a great movie from a grit and determination sense. But it also showed that there is a lot of connection between art and science, and that sometimes you have to make sure that you can have those connections between the two areas to be able to get the best of both worlds. And before I start talking about many other things, I think one quote by Isaac Newton, which I think is a very popular one, at least in the science world, is that if I've seen further, it is because of standing on the shoulders of giants. Now, in the field of genomics, I think, when we today can see a simple report that looks at a lot of different disease, risk and many other things, I think we have to acknowledge and realize that it couldn't have happened if you just had one person who could have thought about all of this, because you basically are, when you look at anything scientific, you look at anything medical or anything else, all of that has been built up upon the work that other people have done. And I think that's an acknowledgement that we all have to make sure that we give, and also make sure that we understand that what we do today, other people will be able to see further because of what we are doing today. So clearly, I think there is a lot that is happening in the genomic space. AI has been better developed today. We are finding that because of AI, you can actually now, when you look at the nanopore sequencers, for instance, the tiny little desktop sequencers, they had initially had a challenge, for instance, of some of the reads and other things, but they used AI to be able to come up with a better overall instrument, for example. The other example is where you can use AI for just in terms of understanding different data types and putting those together, and I'll talk a little bit about how we are doing that. But that also helps you in getting better understanding. Third is someone like Google or any of the places that we consider as for granted. I think all of them have gotten to where they are today because of better AI. And it has helped us, I think, in using genomics has helped us in better diagnosis, in personalizing our medicines, and as COVID taught us, we were able to come up with better vaccines and drugs very, very quickly. But the other thing that you can do with genomics is that instead of spending a lot of time and money on end of life sort of focus on healthcare, I think there can be a focus on prevention, because you now have the levers of what you can change in terms of making it a lot more productive. So I think while there are many more such use cases of where we see genomics and in many areas, but if you have to summarize it, basically you're looking at it for pharmacogenomics, which is personalized medicine. You want to understand if a drug works well for you, both in terms of efficacy, but also in terms of toxicity, whether that particular drug is likely to have a toxic effect for you. The second area, and an area that I'm deeply passionate about, is that of prevention. I think most people think that when it's in your genes, then it basically means that that's it. You have been given a death sentence of some sort. But actually, what it is really a completely different way of being able to navigate, to be able to understand and make sure that you're able to get the right outcome by understanding. In many cases, I think if you think of a parallel, it is about if you start measuring things, for instance, you start seeing that you are able to do better in most cases. For instance, I think when people are trying to lose weight, for instance, when you don't measure it, then it's hard to understand what is going on in your body. But if you do, then and you also measure other parameters, you're likely to be able to make those changes. Similarly, I think when you understand the genetics, then it's easier for people to sort of make the right choices, lifestyle choices, to be able to change the outcome versus not. And then, like I mentioned earlier, drugs and vaccines, I think that genetic information becomes very helpful to be able to come up with not just personalized drugs, but also vaccines such as the mRNA vaccine and others. The other area, of course, has been diagnostics. I think the RT-PCR test that all of us use on a regular basis, I think was done because we had the COVID sequence. We were able to understand the complete, the genome sequence, and we were able to come up with diagnostics very, very early and quickly. And I think we saw that when it came to scale, I think that was probably the biggest and the most impressive demonstration of how quickly science can be developed if you had access to some of the base level information like genomics. And the last, which is actually the more interesting from a curiosity perspective, but also ultimately is related to health, is that of ancestry, trying to understand where we came from and how our whole journey has been across from the last many thousands of years. And this information allows it to be interesting, but at the same time, it is also something that allows us to be able to understand and see what has happened to the human race over the last many years. So in terms of where we are headed towards, we have basically realized that the consumer is the one who's going to be the most empowered. But the consumer also has many different phases of their life that we can address. And I think that initial this thing starts with making sure that by the time there is pregnancy, that you can understand if there is, for instance, you can understand if somebody is carrying a particular mutation, and that if you can sort of see if there are chances of making sure, or at least understanding if what consequences that may have for future offsprings. Then you can understand if you can actually, for a newborn, whether you can actually make specific changes when it comes to diet or others based on that genetic disorder, for instance. Then if you move on to, let's say, understanding of your preventive health, you want to be able to understand where why you are the way you are. For instance, some of it could be behavioral, some of it could be more traits like your vitamins, how you process food, because that's actually a much more bigger area of what you are ingesting versus a medication, really. And also how that has how you react to certain things like exercise and many other such things, as well as understanding if you are at risk for a particular disease or not. And I think that information can help in sort of tailoring or making sure that you're able to get to that. And then we have, a larger and an increasing number of cancer cases that we are seeing. And in order to be able to address those, we looked at two areas. One is to be able to look at it from affordability point of view. And the second was to be able to build it such that it brings more precision in that whole space. So we've built products that have allowed us to be able to integrate different data types, but also we have created products that address specific needs to be able to then bring the cost down quite considerably. So for example, when we built DNA OncoNext, which is an NGS-based product for both tumors as well as blood, I think what we did was we wanted to make sure that you can do a 2000x coverage, but make sure that the price to the consumer becomes very reasonable in the 10 to 15,000 rupee range. The other area that we have also worked quite extensively on is that of drug responses. And we've created a product called MedicaMap. And MedicaMap, we've been able to build on a low-cost platform that allows people to have access to about 150 drugs for a very low, at a very low price point. So this is what we were able to do, that we were able to bring all these products that sort of converge to the consumer. And I won't talk about gbio.me for right now. I'll talk about it a little bit later. Now, when it comes to the whole, Genomepatri and everything else that is there in it, I think it essentially is focused around what we call as the polygenic risk scores. We're looking at many different genes that sort of implicate or are part of the risk factor when it comes to the overall understanding of a disease. And so when we are creating these risk scores for individuals, it is essentially to be able to understand that you have multiple genes that might implicate you for a higher risk, but that it's not the only thing that that is sort of what creates the outcome. But we also look at many other parameters like your lifestyle, your eating habits, your many other such things, environment that you live in, your microbiome, and other things that will sort of be able to make those changes. So we help people understand that. And this is what we do in the Genomepatri, where essentially people don't have to, we look at 3 areas that we can address for, ultimately for better health, which is, can we focus around accessibility? And I think one big challenge is that if you're looking at tier 2, tier 3, or villages and other places, sometimes you might not have a trained phlebotomist who might be able to bring these samples back to a lab in time, for instance. But what you can do is to have a saliva sample that you can look at for a month or 2 and be able to get it back. For instance, you don't need a month, but it's better to be able to do that and at room temperature. So we have an easy way for people to be able to get access to a particular swab. Then we do an analysis for genetic predispositions. We do a genetic counseling, and then we give them a personalized action plan. So this is sort of what we do with not just Genomepatri, but many other such tests as well. So these are the kind of samples that we use. We use either blood or saliva. I think one good thing is that COVID has taught everybody about swabbing. Of course, this is done on the cheek, not on the nose or your throat. So what we look at would be different areas, would be things like your traits. Are you likely to be at risk for alcohol, caffeine, HDL, LDL, triglycerides, lipids, obesity, and other such things. Then we look at things like your disease risk. Are you at particular risk? You can see in this case, there is a population risk, and then there is a genetic risk. And then we have pharmacogenomics, and then carrier status. We currently have more than 100 such conditions. We are planning to add another 50 more to Genomepatri as well. And then you can see this on the phone, web, or a printed copy. Now, on the clinical genomic side, I think there are many areas that can be of interest from any of your point of view. But we looked at about 2 areas that we feel that would be interesting for us. And one that I mentioned, which is the DNA OncoNext, which is a small panel for few types of cancers like the head and neck, breast cancer, lung cancer, thyroid cancer, and so on. And then we have OncoScreen, where we've been able to combine genome, like the information that you're getting from your polygenic risk scores with biochemistry parameters, and to be able to create it as a low-cost screen that is there. And then we are looking at things like exome sequencing and baby map, which sort of looks at both carrier screening as well as newborn screening also. Please feel free to ask questions if you have any. Then we have many different kinds of different targeted panels that are there for head and neck, like I mentioned, lung cancer, esophageal, thyroid, breast cancer, and so on. And of course, any of them that are not done in this panel, we do it on the exome panel as well. Then, like I mentioned earlier, we have a product that is Mapmygenome Match-My-Genome, that allows you to be able to look at recessive conditions for 2 different for parents that are planning a child, and essentially looks at a full coverage across the exome to be able to help people understand, and we counsel upon what next to be done about it. And then, like I mentioned, we also have a product called chromosomal microarray that allows us to be able to look at structural aberrations and copy number variations in chromosomes that will help you to understand genetic disorders, including the neurodevelopment disorders, metabolic diseases, and so on. And this would be an example of how those reports would look like, because it would point to where what you have found in each of these, along with a graph that sort of shows what has happened to each of that, and give a standard response in terms of what has happened, what was the gestational age, what has happened, if there's a pathogenic variant, and so on. we have, I think many of you might be aware, but many of you might not be aware, is that there are 2.8 billion drugs sold per month. But adverse drug reaction is among the top 10 causes of death globally. And 50% of the medicines are inappropriately prescribed at a global level, which means that all of us have a huge opportunity to be able to change this number, because that will allow us to be able to actually change the outcomes for a very large population. We've also seen, for instance, one of the areas that we saw was that Indians are at a greater risk for drug-induced cardiovascular events. And I think part of it could be that we constantly also have, one is off the shelf, but the second is also in terms of we have in certain cases, I think maybe inappropriately prescribed. And in another case, we found that we also have a high frequency of a drug gene variant for fast-acting drug metabolism that increases the chances for adverse reactions. So again, this is again done with a simple non-invasive swab, and we are able to come up with a product like this in about 6,000 rupees or so. So it allows people to understand that, and we are basically saying that this helps you to discover the why, which we initially was in English, which said the why, but since people didn't understand, we added it in Hindi. So this is what we do over there. So essentially here we are using a microarray based product that allows us to be able to bring this to the market. The one area that we are very actively working on right now is where many of the newer students will find interesting, is that we're using a lot of machine learning and AI in combining the different types of data that we are getting. And as I mentioned earlier, we are doing a lot of, now we've started doing biochemistry and other tests. We were already combining Genomepatri and other such products with lifestyle. And now we've been able to add to that other biomarkers to be able to help understand and create a much more clearer report for specific individuals in different areas like cardiovascular, cancer, but an overall health score as well. Any questions so far, or?
Actually, one of the participants is asking when to suggest CMA and NGS to the patients.
Sorry?
When to suggest CMA and NGS to the patients.
So I think it depends on their condition, of course. I think CMA is more chromosomal and copy number variation, and then NGS is you're either typically trying to find a particular solution to a problem. It could be that you're trying to find a mutation that runs in a family, or you're trying to find something that maybe is unexplained. So it's more about trying to find a specific problem on your DNA level. So those would be the two things. But typically the way we would do this is that either your doctor understands that, or your genetic counselor understands your family history, understands where you're coming from, and then writes the prescription accordingly.
Okay, ma'am. You can take the questions at the end also. There is one more question.
Sure.
Would you like to proceed now, or?
Yeah, sure.
Yeah, the question is, what could be the reasons for the increase of cancer cases, mostly among people who are at the age of 60s now? If we see the lifestyle of if there's a good during 30s and 40s, and they doesn't fall in the category of hereditary line also. How could we find an answer for this irrelevant changes in the human genes?
So I think one thing is that cancer is not only caused by genetic, not just your hereditary cases of cancer, right? So it's not that only BRCA, for instance, we find that a large number of, most of the number of tests that are written today are still just looking at specific genes, right? So one is that you want to understand more number of genes that is there. So to understand the hereditary part. The second part is also, I think it is, clearly we are living longer. So that's another thing. But the third and more interesting thing is in terms of what environment we are living in, our lifestyle as well. And so it could be things like, are you living in a very polluted environment? Are you having some of other products that we use? Is there something that is there that is causing these increased cases? I think I feel that a large number can probably be changed if we, for instance, if people understood that your genetic causes are less, not as, if you understand that you have a risk for lung cancer, then if people actually stop, for instance, smoking, that reduces the number of cases that you will see for lung cancer, for instance. So I think it's a combination. It's not an easy answer to give, but at the same time, I think we have to understand that one, we are finding more more cancer cases because we have also started looking for them. We did not look for some of these earlier. So many of people who had comorbidities, I think, if that was discovered first and only identified, then you did not classify it as a cancer. The second thing could be that, most of the time, I think even today, I think we see that more people look at late stage cancers, right? By the time they are not doing an early intervention. But most of these early interventions, if they did, they were likely to be able to actually find that their cancers could easily be either, be treated much earlier than otherwise. So I think there is a number of different things. I hope I was able to answer some of the reasons that we have seen at least.
Thank you, ma'am. There's one more question. What is the demand for these genomics-based products in the market? What is the impact of these products in the lives of people with chronic diseases? Can you please provide the statistics for the same?
Sure. So, so I think clearly, I think the demand for these products is increasing quite rapidly. So there, there is the clinical part, and then there is the consumer part, right? So the clinical part, I think, one, as the number of doctors are understanding this more, we are seeing that we are finding that there are many more times that clinical cases we are starting to see a lot more in the market today. I think there are many more companies in the market as well. So that is one part. The second part is to understand it from a consumer point of view. And I think here is where you can actually make the biggest difference, because if you look at, for instance, cardiovascular diseases, 80% of those are preventable, which means that the largest number of deaths that we see in the country today many of them are preventable. So I think it has a very, very significant impact, but people have to know about it earlier rather than later. So if you are able to understand and make those changes in your lifestyle ahead of time, while you are 30s and your 40s, not wait till you're in your 60s and 70s, then I think you will see the biggest impact. The second, for instance, is if you look at diabetes, diabetes is not one problem, right? It sort of is a systemic problem that is there. But if I take my own case, strong family history, my father had diabetes at the age of 40. Pretty much everyone in the family had. But I think when you are aware, you are likely to make changes. You make sure that you exercise regularly, you eat proper, and you are constantly also measuring parameters that you can sort of help to understand what is happening with the disease. And I think with that, you can actually start seeing that, I don't have diabetes, and I was able to actually reverse some of even the trends that we were seeing with HbA1c and others. So I think ultimately, I think if people understand that if you know this better earlier on, then you're able to make changes that has a very large impact. So diabetes is about 60% and cardiovascular 80%. Actually, cancer is also, studies have said that 50% of the cases are preventable. So I think if you just look at these statistics, they are quite significant when it comes to what you can have as an impact for the population. But if you are waiting till something happens, it means that you have waited too long. And by the time, I think it becomes harder to be able to make changes. You can still make them, but it's more that it becomes harder and more expensive towards the end of life rather than earlier.
Yes. So there's one more question from Dr. Ajit Kamath. He's here. Sir, would you like to unmute yourself?
Hi, Anu. Hi. Thank you for agreeing to do this talk. Very interesting. My question is related to liquid biopsy. Can you please tell us how things are in India? I think it's, it's at least in the US and Europe and all, is becoming kind of, mainstream for cancer detection, diagnosis and other things, whereas in India, it's not much. So I just wanted to get your thoughts.
Sure. So I think, when it comes to the GRAIL type of detection, early detection with biopsy, I think obviously we don't see too much of, which is basically finding it in very early stages. But I think we are starting to see that for somebody who has been operated, and then they do that to understand if, what the possibility of recurrence or remission would be. I think we are starting to see those pick up significantly for some of the cancers. So I think now that from a technology point of view, I think it has become more standardized. We are starting to see that. But at the same time, I think it is also a matter of cost. I think the cost is still, still a little bit high. And I think maybe once it comes down a little bit more, we will start seeing that. I think, if you want to get something like GRAIL, then that has to be done ahead. So the moment you know that you are likely to have some sort of a possibility of being, because of your family history or otherwise, then people should be able to do that upfront. So that I think is still not yet there, and I think it'll take a few more years. But yeah. I think the access to technology is there today, right? Much easier. The bigger, a lot of the companies are creating these kits to be able to create these, liquid biopsies. So I think that part of it is easy. I think it is awareness and it is cost. The two areas that I think we need to still address further.
Yeah, but Anu, the cost is still very good in India compared to US and Europe and all these things. Still, it is it hasn't been kind of adopted by the clinicians and others. I hope it happens soon for the benefit of the patients.
Okay, I think it is happening. I think now there is a lot of market awareness, I would say. But I think it is, I think one thing that I don't know how we can, maybe as FABA can address this, is how do we get some of these newer technologies to the physicians, right? In a way that is easy for them to understand. Because clearly a lot of these physicians and oncologists and others are quite busy in their practice. And it's hard to be able to impose on them newer technologies without making it in their language, if I would. Right? So many times I ask some of these oncologists in terms of, how can we help them really? And one of the things they told me is, you tell us, clearly, simplify it for us. Right? We don't want to see a complex report. We don't want to see that, likely something is there. Because it's hard for them to understand. So I think if we want to see this happen in the clinical space, we have to make it simpler for people to sort of take those next steps. And I think, if I look at many of the reports, I think it still is, we want to show the science, but actually I think we need, if you want science to be there, I think it also needs to be communicated. So I think we have to do some work on our side as well.
Yeah, thank you. Thank you.
I think to add to the point of Ajit, that exosomes are good biomarkers for many of the diseases, including cancer. So isolating the exosomes and then sequencing the various RNAs, microRNA and many others could be a very good way of detection, early detection of various diseases. So I just would like to know how, whether this is being at least thought of at Mapmygenome or Ocimum.
So I think, it definitely is being thought of. I think the bigger challenge, of course, is, education. And I think, and I think one of the things that I'm trying to do with some of the physicians, I don't know if you have seen any of that, but I'm trying to get physicians and try to understand their understanding of genomics, because if we can simplify it, then maybe we can get them to understand and see that there is a tangible value proposition for them, right? And I think if we are able to communicate that, then we will see that happening. So from a research point, yes, we can do it. I think, how do we build it into something that's commercial? I think that requires more than building the product. I think that's, and I think both what Dr. Ajit, you have mentioned, I think that gap is still there in terms of translating that to normal clinical practice, and not for a medical geneticist and not for a specialized person, but can we do it for, you know, a regular oncologist in a tier 2, tier 3 town, or somebody who's a general practitioner, right? And I think that's the same problem. I think even in the US we face that that there are still many tests that are not yet have as much of an uptake, and part of it is they've not been able to see real value, and they're not able to see real value because we've not been able to communicate. So I think that gap is still there.
So that points out that FABA has to address the problem of research development and clinicians. So whatever the gap that exists between the two, I think we need to have some webinars focusing on that point.
Yeah, so I started doing these genomic gupshups where I call clinicians and others.
FABA will be happy to join that and then take it up further, not only with you, but even other
I think every
developments also.
Collect with all the different companies have different initiatives going on. I think if we can get it together, I think that will be a fantastic place for people to see how to get access to that education in one place.
Very good.
Thank you, ma'am. So there's a Nigerian participant who's saying that he is interested in preventive use of genomics, and asking if you can emphasize on this, especially in view of your experience in COVID-19 pandemic.
Sure. So let me see if I can take it to the next few. So these slides, a few were on I actually think I might have removed some, but I'll talk a little bit about what we did during the COVID pandemic. I think when we look at COVID, when we looked at the whole COVID pandemic, I think one of the areas that we felt that we could actually help people was to look at the data that was there from a immunity perspective. And so we analyzed the data that we had by looking at information that came from the previous SARS and others. Let me see if I can get to that. Yeah. So what we have done over here was that when we looked at Genomepatri immunity, we looked at a few things like nutrition that specifically might be impacting cytokine storm, comorbidities like your diabetes and cardiovascular, and we looked at the SARS-CoV-2, not COVID-2, but SARS-CoV susceptibility. And it was interesting because, there are and some parameters like, other parameters that were included in this. And what was interesting was that, and I shouldn't say this yet, but, so far I've been lucky. I didn't have COVID yet. But on the report also, it sort of showed that. And some of the people who did have higher susceptibility and got multiple times, I think you could also see that in terms of that correlation. So it was a reasonably good correlation between what you could see in the Jiva studies and what you could see here. Of course, there are many other parameters like, have people been careful from using masks and various other things, because you can't obviously depend only on your genes to protect you. But it's an interesting study from that perspective. And nutrition is an important element because I think it's people don't give it as much importance, but that can actually have a big effect in terms of how your body reacts and creates the right kind of immunity for you. So I think that also helped a lot of people to understand that. I hope that was what you asked.
Yeah.
Yeah. Okay.
So there's another question: how environmental pollution affects the human genome and genetic disease? What should we consider before giving genetic products to a particular patient?
Okay, so environmental pollution and other things, right? They obviously affect not the human genome, but your epigenome, right? So your base level code is not changing, but how it's expressing itself will change. And I think part of it is that when you, over a period of time, of course, we might see that if your environment has changed, I think you also find that over a long period of time, based on what will survive over thousands of years, those will be there. And I think some of the examples of those we see today in terms of people who are better runners and all of that, but that has happened over thousands of years. But pollution clearly plays an important role, and we can start seeing that if you start looking at your epigenomics and looking at what is happening with the different methylation and other things that are there. But it's not actually changing your hereditary code, at least not for your own generation. But over a period of time, we'll see the ones that will survive in a particular area will be those that might be able to actually live through those environmental changes over thousands of years. And what should we consider before giving genetic products to a particular patient? So I think here, one is that, if people are using it for, what is the purpose for using a genetic test, right? That might be very different for different people. If someone wants to find their ancestry, I think anyone can do it, right? I mean, if you want to understand if you're 30% Punjabi and 40% Rajasthani, I think that is information, I think, easy for anyone to digest, for instance. The second could be like you want to understand nutrition. I think again at any ages that you can understand that. When it comes to diseases, then it depends on your when you want to understand that, right? So if you are a 15-year-old, maybe you are not that keen on understanding if you're going to have Alzheimer's and Parkinson's or cancer or other things. You might want to know more about nutrition and fitness and all those other things that are making you a little bit do better in whatever you're doing. But I think as you get older, people will understand that, they will start seeing families around them. And I think that's when we see a lot more people doing more on the disease risk. And then the ones that come later on would be where people will try to do a pharmacogenomics profile and keep, because if for any reason someone ends up going to the ICU, I think that information can be very helpful. But none of us know when we will actually end up going over there. So having a pharmacogenomics profile can be very, very helpful. But again, it depends on consumers that is there. So these are all what we consider more as something that would be triggered by the consumer. And then there are others that you want to understand the specific reason. I think those typically will come when a doctor thinks it's necessary to understand either the reason behind it, or they are trying to understand how to treat that particular disease. So I think those are typically either comes as a result of talking to a genetic counselor or as a result of talking to a medical doctor, that you will find that specific products would come over there.
Okay. Suma is asking, does this genome techniques help with any cancer diagnosis and treatments?
Yes, absolutely. I think, see, cancer ultimately is a genetic disease. And so two things again. One is, especially in the case of cancer, there is, one is to understand if you have a inherited risk, right? So what we call as the hereditary, for instance, the hereditary cancer panels are typically to understand if you have, for instance, the BRCA gene mutation, you have some other gene mutation that is sort of putting you at risk higher than the average population. So that is what I would consider as your hereditary risk that is there. But then there is the second part, which is that you already have cancer and you want to understand the treatment for it. Then typically we would take, for instance, your tissue samples and others to be able to understand and sequence those. And there you would do it for, let's say, 2000x coverage or 1000x coverage to understand if there are any driver mutations and other things that will be helpful in creating the right kind of treatment platform for that specific patient. So for cancer especially, I think these have become extremely important to understand, because some mutations will make certain drugs not work very well, and some mutations will make some drugs work well. So I think there are many areas that you can use genomics in cancer, and these are just some very overarching kind of tests that are available for cancer diagnosis.
Yeah, there's one more last question before we proceed, ma'am. Are there any large-scale studies done where the genomic-based information have been shown to be effective in terms of better treatment and prevention?
Yes, there are actually hundreds and hundreds of studies that have shown that. For each disease, you'll find that there will be many such studies that are done. So I would suggest that if you're interested in specific areas, go to Google Scholar, search, and you will find many such things. So for instance, if you're looking at has it been effective in better treatment for diabetes or cancer, anything, or a specific type of cancer, I think you'll be able to find that. We'd be happy to send you some if there's interest in understanding some of these. There are also interesting papers that have been done. That's a good question. I will respond to that. But there have also been, now I've forgotten what I was saying. Yeah, so I think what is also is that there are also studies that have shown the value of consumer genomics for better treatment as well. So here you said in hospitals, doctor tried drug directly and we are not informed about this kind of sequencing. It is available in India. It's available with Mapmygenome and with many other companies that are there. And I think there are two reasons doctors don't always tell you about it. One is that they want to not be able to understand and explain to the patient. That's number one. And the second reason is that they don't want to burden a customer with more tests, right? So those could be the two reasons that they feel that they are not burdening someone, but actually if they understood the value, then they will probably do it, because at the end of the day, I think then it's a better return on investment. But I think that's the sort of education that we are talking about. Even Dr. Ajit and Professor Edana, we were discussing. I think it's exactly the same problem that is there. That usually this is not always being asked, being sent. But I think today, if you go to some of the cancer hospitals, I think sequencing is definitely part of the solution that they are providing.
Thank you.
Dr. Anu, if I can just add to that, I think it's a very relevant question. What happens in most of the hospitals is, larger hospitals, as you rightly said, they have molecular, oncologists with experience on doing mutations, characterizing them, and telling them, which drug is available for what type of mutation. But other hospitals do not have it. This again, for the question that's asked. So the doctor will just try exactly the same what he or she has tried in the past for similar patients. Okay, so they would rather go with that rather than taking some biopsy samples, sending it somewhere else, and asking for the panels to be done, or 200 genes, 300 genes to be done, and it takes quite a bit of time and it is expensive. So the doctor in a regular center, smaller centers, will just go with doing the chemotherapy and all. But I do know, when I talked with many of the oncologists, the doctors find this as magic. Okay, when they find some specific mutations, and all they have to do is a single tablet a day, and that's it. The cancer is under control. So there are a lot of opportunities, particularly, and all the hard work that you're doing to, make this accessible to the patients. I think it has immense value for the patient care.
I agree. And I think what we've started doing is that we are now getting a lot of queries directly. People reach out to us directly. So we usually, what we are offering is genetic counseling to the physician, to the oncologist, so that in case they don't have that expertise needed, I think they are, they can reach out to us. And I think the information build has to be from both sides, right? The consumers being more aware that, there are such options. And the second is, can we also bridge the gap between with the oncologists as well? I think that's what we are seeing. So that's why typically when we look at some of the sales, for instance, numbers that are coming in, it's usually the same set of doctors that are prescribing these tests, because they see value and they are constantly doing that again and again. but not always from some of the others. So I think that's an area that we all need to pitch in and bridge.
Yeah. Thank you.
Yes, ma'am. There's another question. Why genome analysis method cannot be mandatory before diagnosis?
I think it's a good question. I wish it was, but I think there are two reasons for it, right? I think one is that it has to be, when you look at healthcare, I think if only about very small percentage is currently paid by the government. I think we all are paying for it on our own. So actually, we can demand that sort of an analysis. But I think part of it could be that you don't want to burden everyone with tests that maybe they might not be able to afford. That could be one reason. I don't know why it is not yet there. The second is that now that the costs have come down quite significantly, I think people are still not aware. Right? So what in the last few years, we are seeing that suddenly even like your NGS tests have come down quite dramatically, and they will continue to do so in the next few years, some more years as well. So I think the cost will become a matter that will go away. It is no longer the 50,000 and 55,000. But today I think someone just asked the price for a genome analysis test. So when we are doing a 2000x coverage for a small panel, for some of the ones that we have, there the end user price is about 15,000, and which is not much at all, because it used to be a few lakhs. And I think that was the reason that when you look at foundation and others, I think it becomes unaffordable for a large number of people. The second is that when you look at it from a risk, the preventive test point of view, our typical prices are between 6,000 and 6,500. That's the range that we are looking at. And I think the NGS tests, ultimately, they are currently, if you're looking at the whole exome, not for cancer diagnosis, but for other these things, I think typically we are finding that the price point is around 20 something in that price point. So the changes have come quite significantly. I think we need more lower cost, high quality tests, because the doctor will start seeing the real value for what is happening to their patients, and as a result will become a good cycle in terms of the number of people wanting to do it and seeing the value of that. So I think those are probably the areas that we can still improve upon.
Thank you, ma'am. You may continue with the presentation. Yes.
So I think I had, I've already taken an hour, so I don't want to take too long. But I think this is an interesting part, which I think is not disease focused, but this is more about understanding, where we have come from. And so we started doing this not very long ago, but it was a product that you can see over here, that this person actually is from Kerala, but you can find that 24% of his genes were from Pakistan, about 17% from Pathan and Sindhi and so on. So you can find that people are actually quite intermixed when it comes to, when we look at the genes, and that we are not very different from each other. But this is a great place for people to have that initial conversation about, how you can, what has happened with our genes and what can be there over here. Then I thought that one more quote that will be helpful to understand is like, the genome is a book that has written itself, continuously adding, deleting, and amending over 4 billion years. So I think what we see today has been multiple times, many volumes of those books that have been written. But what is interesting is that if we can start analyzing that, we can also understand what happened and where our version 1 was published, if you will. Right? So we can start to understand what has happened to us over the years. So I think if we just, could just, little bit summarize, I think you can, one is find susceptibility to disease or past predispositions of your body. You can understand body fitness, nutrition, fitness that is encoded in your DNA, and roots and ancestry, and understand, if there are clinical problems. So in many ways, it's like holding a treasure map to the golden standards of life, right? And they are right, that is there inside of you. So I'd like to end that the future is now, and we don't have to wait for it. I think it has been a very nice and interesting conversation with all of you, and happy to take on if there are any other questions.
Thank you so much, ma'am. That was a wonderful talk, and you have covered all the basic details for students and everyone who are interested in genomics. And there's one more question from Priya. She's asking, could we get any hands-on courses for students or PhD scholars? I think this is for FABA. Professor Redana is here. To get actual scenario and how to use genomics in real life.
I think we have been conducting such kind of courses, maybe in association with so Dr. Anuradha Acharya, we could probably come up with some courses so on a specific topic. Last webinar was an miRNA. I don't know what is the RNAi technologies, how they could be used in agriculture, and RNAi technology could also be used in human, health also. So we have been conducting such kind of webinars that maybe we could probably plan some of them specifically for the student purpose. Sure, Monipriya.
Suma is asking if she can apply any projects in your company for her MSc.
So I don't know. I mean, I've been thinking about some, even for people who are within the company, to see if there are possibilities of doing a PhD. I don't know about MSc. Maybe we can have a longer discussion and see if those are possible solutions for us to be able to do and how to implement them.
One question, Anu, so are you getting the enough right candidates for the data analysis that you get when you go for a complete human genome sequence?
Yeah, now that's not a problem. I think we have
large number of data scientists.
We have, so we've got now, one of them came from an academic institute, recently joined us, came from a statistical background. We have Dr. Sandhya, who's been with us for more than 10, 12 years now. So she's been able to build that team. But of course, I think we want to continue to enhance that team because we're trying to put together the genomics and the other data types as well. So we would be looking at getting more people in that area also.
So in fact, FABA conducted a couple of maybe 2 or 3 workshops on data science, data clinic also. So physical workshop that was, and a couple of them were online workshops.
There's another question, how to develop industry-ready skills? The intakes for life science companies is comparatively less. So what can I do to increase my employability?
So I think one is that if they do try to see if they can get projects in some, like an internship or something in some of, current companies that you can do. The other is that there are also other courses that you can do which sort of are more applications oriented, right? So if you look at, Coursera and a few edX and a few others that are available, I also continue to keep doing my own, there is one, for instance, that I'm doing right now called Precision Medicine, and it's by the University of Geneva. And it's very good, right? Similarly, there are courses on diabetes and many other such courses. So after you're done with your MSc or anything that you're doing, some of these can be used as supplements for what you can be doing also. And that sort of helps you in getting access to information that otherwise maybe wasn't part of the syllabus for your college. So that one of the things. Second is, I think, read more books and try to do some research that will allow you to be able to come up with something that is more interesting. Keep tab of what's going on across the world by looking at publications, and I guess those would be the areas that I would recommend. And if you can get access to internship, I think there's nothing like it.
Monipriya is asking what should be the minimum essential qualifications, work experience for a person to enter genomic study industry after their master's?
I think it depends on what you are entering as, right? I mean, so for instance, if you are taking somebody as a genetic counselor, then typically we'll take somebody who's already qualified for that. Right? If you're taking somebody for, let's say, being part of the lab, then I think usually the team will not let you do a lab immediately unless they'll give you some trial runs before they will take you in. So it shouldn't, after their masters, typically I think it is more about making sure that you can be in some place where they can watch how good your skills are and then take you. If you're looking at it from an analysis point of view or bioinformatics point of view, then I think, at least we take mostly people who have a little bit of experience, but I think that's a place, for instance, some of the companies might need many more people. They're more likely to be able to get you in. You probably will start with simpler problems to solve and then move on to more complex issues that you might be able to help them.
That's it, ma'am. Thank you so much. You're very kind enough to answer every question in detail.
Sure.
And you have enlightened us with genomics and about Mapmygenome as well. Thank you so much again on behalf of FABA. Redana sir, would you like to take? Ajit sir.
Yeah, thanks,
yeah.
very much, Anuradha, I think, for sparing your time. And you also have given ideas where we could work together. One is, anyway, in terms of bridging the gap between the scientific developments and the clinicians' understanding. Definitely, we will discuss, maybe myself, Ajit, and you, and many others will have a kind of brainstorming session and then come up on the topics where we could have such kind of workshops. And that's one. Then in terms of the training programs that we can probably do, we have done such kind of training programs on data science, and we also have done on molecular modeling and drug design. But on genomics and specifically what it can offer, maybe we could design a course for the benefit of the students in maybe near future.
Yeah.
Ajit?
I just want to add, thank you very much for talking to our students and other participants. I think you have been spearheading this effort to bring genomics in India, making it accessible. I can't thank you enough because people are risk averse taking such steps. But it's nice that you thought about it 15, 20 years ago, and now genomics, sequencing and everything, particularly for diseases or rare diseases and everything else, is becoming accessible in India. So thank you for the nice talk.
Thank you so much.
Thank you.
For everyone, there's an link in the chat box. Please fill the form for the certificates. This window will be open for another 5 minutes. Thank you, ma'am.
Yeah, thank you. Bye.